Okay. It's 9 o'clock sharp. So hello everyone. Welcome to today's Zoom seminar. And again, I want to have special thank for our speaker, Dr. Yifan, to spend the time at this late hour with us. And then obviously everyone online be with us today for the weekend and this late hour. So it's my really pleasure to welcome our today's speaker, Dr. Yifan from UPenn. He currently is Associate Professor of Radiation Oncology in UPenn. So he actually finished his MD's training in Shanghai University of Traditional Chinese Medicine, and then continued to pursue his PhD training in pathology in Case Western Reserve University School of Medicine and in Ohio. So after he completed his PhD, he moved on for his postdoc training in Department of Cellular and Molecular Medicine in Cleveland Clinical. And then from 2014, he actually started his own independent research program as assistant professor in Department of Radiation Oncology in UPenn. 2020, he was promoted as associate professor with tenure in radiation oncology in UPenn. So the main research interested in Yi's lab is focusing on a couple of different aspects, including brain tumor, angiogenesis, tissue repair, immunotherapy, cancer stem cells, and proton radiation. So his lab is working on cancer and regenerative medicine as a predominant example of malignant solid tumors. Glioblastoma is among the most deadly human malignancy, largely due to its high resistance to standard therapies. So the ultimate goal of Yi's lab is to develop efficient therapies against disease by targeting the tissue microenvironment, which I think we're going to hear a lot of today, particular vascular remodeling during the tumor immunity. And then Yi's lab also aims to reprogram the microenvironment to block cancer progression, activate anti-tumor immunity, and fuel post-injury tissue repair. So Yi's lab is extremely productive and then generate a lot of high-impact work. And then based on these achievements, he was awarded with numerous honors and awards, which including the Young Investigator Award of Excellence in UPenn, Sharp Award, National Brain Tumor Society, and then Early Career Investigator Award, Radiation Research Society, Innovative Project Award from American Heart Association, and Springer Junior Investigator Award from North American Vascular Biology Organization. And then from 2019, he got Research Excellence Award from American Heart Association. So I think today you're probably going to tell us a lot of, as I just mentioned, vascular regulation of tumor immunity. So with that, I want to thank you again, Yi, to spend your time during the weekend with us to share your science with us. Really appreciate it. Please go ahead.
Thank you so much, Trent. So I think this is such a big honor and pleasure for me to present our research. And I think this is a very comprehensive introduction. So I maybe save a lot of slides for me. So I just focus on our research and science. So actually we are working with cancer. So when we talk about cancer, I think we are super lucky. So because right now is really exciting time. So I think we can right now can dream something extremely big that we never can thought about in maybe just 20 years ago. This is maybe in our lifetime we can see something big thing happening, and then maybe can real cure a cancer. So that is a dream. So eventually come to true. So this really exciting thing is everybody knows is immunotherapy. So actually, when we talk about immunotherapy, what I was talking about to our graduate student, I'm teaching a class at the Penn, so that is cancer biology and genetics. So immunotherapy turns out is one of the oldest therapeutic approaches against cancer. It's been, I mean, since I would like to say we in college developed this technology in back to, I would like to say this 100 years ago, even is older than most other treatments. So, but right now we can see this is a fast growing field, especially we're talking about right now there's two star approaches. One is CAR-T, another one is checkpoint blockage. So immunotherapy is just like how we can start a car. So actually when we start a car, and the first thing is we turn on the engine and release the brake. So one thing exactly happened to the cancer immunotherapy is the checkpoint blockage just work as a release the brake. And then CAR-T will drive this T cell become active, just like make this engine become active. So, but this is still a big problem, even though we can see lots of really exciting things happening. But if you look at the general solid tumor, you still see less than 10, or maybe we'll see that definitely 20% of patients are not so response to the all kind of immunotherapy. For example, like lung cancer, well, I say only 15% of the patients respond to checkpoint blockage. So actually in our department, we did some like first CAR-T immunotherapy clinical trial in GBM in the patient with brain tumor. So the very sad thing is we tried almost 30 patients, almost none of them, had the response. So the major problem is when we check the pathology, we can see almost T cell infiltration, or see less than 1% T cells eventually get into the tumor. And also for this 1% T cells, these CAR T cells, majority shows Treg phenotype. That means they are inactive. So this brings us a big problem for the solid tumor immunotherapy. That is, first one, the tumor itself is very immunosuppressive. That means T cells will not have very little chance to get in and also become active. And the second is we're dealing with is because this tumor vasculature extremely abnormal, and then T cell will need this vessel to get into the tumor. So in our lab, we specifically working with two fields. One is the tumor immunosuppression. So we work very hard with myeloid cells. That's the major source for tumor immunosuppression. We work with macrophage, MDSC, and dendritic cells. We're trying to reprogram the immunosuppression, and then we'll make the T cells become a little bit happier. And the second is the tumor vasculature. So we want to open this gates for that and let the T cells to get in, either endogenous or actual exogenous like CAR T cells. So make them become, get in the tumor and then start to kill the tumor. For the sake of time, we're gonna focus on the second part. We're talking about how we thinking vasculature can be work as a major component for the tumor immunity regulation. So when we talk about the anti-vascular therapy, we have to mention Dr. Judah Folkman. So this is our hero and the pioneer, scientist for that who actually bring up the whole fields back to 1970s. So his first idea is because tumor vasculature is very critical for the nutrient and oxygen delivered into the tumor to support the tumor growth. So why not we can shut off the vessel and then we'll stop the tumor to death. So, but unfortunately, this is really, even though this is really exciting theory, but unfortunately it did not work so well. So actually people already tried over 20 different solid tumor types, but almost none of them worked with, shows really good promising results for the anti-angiogenic therapy. For example, using bevacizumab, the famous anti-VEGF antibody. So this lets us rethink about and how we can really, push this field forwards. One particular thing is because once you shut, because vasculature is very important for not only for the nutrient and oxygen delivery, but also for the immune cells division the tumor. If you cut off the pathway, and then we'll expect maybe we'll block the T cell infiltration. That's something we will not want to see. And second thing is tumor vasculature is always abnormal. So that's what thinking about another idea is, instead of killing this vasculature, but how about we can normalize vasculature and then let more T cells in, and also maybe we'll let more drug get into the tumor. For example, I will make this tumor more sensitive to the chemo and radiotherapy. So based on this idea, another hero, Dr. Rakashian, proposed another really exciting, theory, that is vessel normalization. So again, so the idea is maybe we can normalize vasculature to improve the tumor, response to the treatments. So his idea is the major driving factor for the vessel abnormality is because it's the imbalance between anti-angiogenic and pro-angiogenic factor. Again, is VEGF, too much VEGF will drive this vessel abnormality. So his idea is using low dose bevacizumab, we maybe can normalize vasculature and then improve the tumor response. But unfortunately, this theory again is very exciting, but therapeutic efficacy in clinic is not so good, and we only can see small effects, and also the effect is very transient. So based on our recent study, we're trying to propose some like a new concept, that is, instead of thinking about this extrinsic growth factor, when we think about the intrinsic. And we believe this vascular abnormality is not only driven by this angiogenic factor, but also driven by this robust endothelial plasticity. So when we look at the embryonic development, you can see endothelium has a very robust ability. They can even form all kinds of like different cell type, even for hematopoietic cells and also lots of mesenchymal cells. And then our study actually we worked this back to like 10 years ago. We put all of our early study together. We want to try to propose endothelial cell plasticity mediates cell transformation, maybe drive vascular abnormality, and eventually will induce lots of bad things happen to tumor immunology and other phenotype. So first idea is this tumor endothelial cells acquire some mesenchymal phenotype, and then to drive the endothelial cell become over-proliferative and also migration ability is high, and then eventually drive this bad vasculature, and also to lose this original endothelial specific gene expression, for example, like VEGF pathway, and then make these cells become resistant to bevacizumab treatments. And finally, these ones is all generate local immunosuppression, and then will make a kind of immunosuppressive niche. So the classic idea actually is not like this. Actually, 2 or 3 years before, majority is from the Dr. Rakul Karori. He proposed actually, he actually observed very similar phenotype. Actually, when he checked the tumor vascular, he can see the vascular endothelial cell express some mesenchymal gene, for example, like FZP and alpha-smooth actin. So his idea is this endothelial cell de novo generates tumor-associated fibroblasts to promote some like immunosuppressive phenotype. But after the careful examination, so actually we found actually this endothelial still remains the key vascular function. For example, like they can remain the uptake of the lipids, and also they can form the vasculature on the medulla. So we think maybe we can still use endometheline, and traditional ideas endothelial mesenchymal transition, but we'll do a little bit of modification. We'll name it after like endothelial mesenchymal transformation to emphasize actually there's no cell fate change. They are still endothelial, but acquire some really bad phenotype to generate the best vasculature and aberrant vascular niche. And our further study shown actually this transformation is so robust, and then they were across lots of different phenotypes, including mesenchymal phenotype and also stemness phenotype. Because mesenchymal phenotype is always associated with tumor immunosuppression, because this has been well established. Tumor-associated fibers can secrete lots of anti-inflammatory cytokines. So this lets us to think about one question. Maybe they were a kind of like a vascular niche. They will serve as a source for the tumor immunosuppression. Because and one of the challenges when we publish a paper and when we talk to other people, they always challenge me because they were thinking maybe this is not so important, because usually tumor and cysts only account for 5 to 10% of total tumor mass. So how you think this is very important things? So our answer is maybe this is local education facility. Because think about all these lymphocytes, once they need to get into the tumor, they have to pass this local vascular niche to get in the tumor. And then this local vascular niche will secrete something. I think this is local enrichment mechanism, and then we'll educate this cell. And then we'll think about, this is our big arguments. We think vascular niche is. So we work with several different cell types. One is starting with working with myeloid cells. So here is one example. When we look at this macrophage polarization, so by the way, we're working with brain tumor, and actually the myeloid cells, especially macrophage, as a major source for the tumor immunosuppression. And very interestingly, we will see the immunosuppressor type of macrophage are closely localized with the vasculature. And when we look at the human patients, you can clearly see the macrophage is associated, with the tumor and the cyst cells. And then we will continue work as we dig out actually endocyst-derived IL-6 as a major driver for the macrophage immunosuppression. Before us, people believe that IL-4 is the major M2, alternative macrophage M2 polarization in cancer. And actually we shown that IL-6 can equivalently have the equivalent potency to drive the M2 immunosuppression. And then this is detailed back, and it turns out were active with M2 and then PPL gamma, eventually leading to the arginine expression. So arginine, as everybody knows, is a very important immunosuppressive, cytokine molecule they can consume and take over the arginine, and then arginine is a very important amino acids for T cell activation. Once delete this arginine in the tumor microenvironment, T cell become inactive. So that eventually drives the tumor immunosuppression. So based on this study, actually we want to think maybe this is really good target for us. But very unfortunately, when we really trace, our animal model, we have used so many different models actually, but we did not see very robust efficacy. We can see animal, get like a little bit more survival and the tumor smaller, but definitely we did not see very robust phenotype. We never cure any mice. So that was lets us think of maybe there's several additional hit we should think about. And then we did a lot about bulk RNA-seq and single RNA-seq, and eventually figure out this is the parallel pathway for the IL-6. So IL-6, even though they can drive the M2 polarization, but turns out that they can also induce CD40 expression in macrophage. CD40, everybody knows this is a co-stimulant factor. So otherwise, IL-6 has a double role. One is positive and another one is negative. So based on this result, we try to propose maybe we can combine these anti-IL-6 and pro-CD40, and then we can do the further cancer immunotherapy. So that exactly the magic happens. So that actually in our lab is something we can see is really amazing. So we treat this with our animal model. We can see this double treatments plus checkpoint blockade can pretty educate all these tumor, and then eventually we cure all these mice. So this is something we never saw it before. So frankly, I have to say this is only one model. When we see another model, it has effects, but not like this. But this actually inspires us a lot. So the lesson is, if we can do some really well designed and immunotherapy, maybe this some way we can really cure this disease. Actually, by the way, I have stated glioblastoma is one of the most lethal human disease. So that is like something we can see in the lab. So the really another exciting thing we can show is right now at the Penn, we already initiated the clinical trial. We already treat, believe is more than 10 patients. So far, we have see something really promising. We'll see maybe 2 or 3 years later, we can share more some like a very exciting data with you. So based on this data, we're thinking about anesthesia maybe it's critical for the tumor immunity regulation. So there's several things we have to emphasize. First thing that I just mentioned, they can form a local immunosuppressive niche, and then they can inhibit the T cells. And second thing we'll have to see is, so can we really reprogram these endothelial cells to normalize the vascular structure? And there's another thing we'll also think about is, when we look at the tumor vasculature, so these three bad things happen to this vasculature, and one of them is the endothelial start lose lots of adhesion protein like ICAM, WCAM. So that's why T cells have a very little chance to attach to the endothelial cells. So that blocks the T cell infiltration. So we were thinking maybe we can find a good strategy to reprogram the endothelial cells. We can break these three major barrier, and then we'll really make this immunotherapy effective for the cancer treatments. So because this is a new field, we have no idea about how this pathway is regulated. And this is a transformation and this is a plasticity in tumor microenvironments. So we started looking two kinds of different screening, so done by two very talented graduate students. So one is we focused on the whole kinome because we want to really find a therapeutic targets. So everyone knows, so the kinase actually is and also receptor is almost 90% of our current drugs. And second thing, we also did a whole genome sequence. We wanted to build the whole regulatory network. So, because recently we already published this first part, well, maybe you can talk about the first part, the kinase screening. So basically we isolate and system from human patients and put two genetic probes. So one is the alpha smooth muscle actin promoted during fibrous luciferase. So this is kind of report for the mesenchymalization status. And then meanwhile, put CMV granular luciferase as a kind of basal control for the basal transcriptional activity. And then on the top of this, we put shRNA targeting the whole human kinome. So this is one by one. So this, even though this is one figure, but our grad student took almost half year to finish this, because for each kinase is 5 or 7 different shRNA, and totally this about 300 or maybe close to 4,000 assays. So eventually we put everything together and we found PAC4 turns out is on the top of the list. So what's the PAC4? PAC4 actually has been known as a major kinase that responds for cell migration, and they can get some signal from the growth factor, and they eventually make the cytoskeleton reorganization and drive cell proliferation. And in our hands, we found this is something, new regulator for the cell plasticity. So this is something we'll call this eye-open figure. So this really make us to believe PAC4 is a new regulator for vessel abnormality. So if you look at the top panel, this is a typical endothelial isolate from the tumor. So this endothelium doesn't look so good. It look like a fibrous look like phenotype and morphology. And then once you put them on the matrigel, they will not form a good like a blood vessel. But once you lock out this gene PAC4, and you can see they can pretty change the endothelial morphology. They can form a really good, like beautiful endothelial structure. And once you put all them on the matrigel, they can really form a beautiful blood vessel. So based on this, we'll want to explore what's the regulatory mechanism, how PAC4 regulates and reprogram the endothelial, genetically let them become normalized. Once you lock out them. So we did a biochemistry, and then very interesting, we turned out PAC4, even traditional people believe this is a kinase that is responsible for the cell migration. Turns out they can reprogram the whole transcriptome, and every gene has been shifted. And more exciting for us, once especially look at the adhesion protein, something really interesting happening. So that is, they can let, once we lock out PAC4, they can let endothelial react lots of adhesion protein, including ICAM, WCAM, that is responsible for the T-cell adhesion. And also there's a lot of clouding, because the clouding actually, this is a tight junction at the protein, and then the hold the endothelium to each other. Once then the tumor endothelium start losing endothelium, they will make this endothelium dissociate from each other, and they will eventually generate the really bad vasculature. So the next step is we want to look at what's the phenotype in vivo, and then we generate endothelium specific PAC4 lockouts mice, and then we'll see this. Once we lock out PAC4, I want to emphasize this, we only lock out endothelial cells, but we can see dramatic phenotype in vivo. It actually turns out the tumor become much smaller than this control, and also animal has much better survival. So this is likely when we check it with the immunophenotype, we can see lots of T cells. And the more exciting thing we will see is, we can see the endothelial cell in vivo endothelial cell, they can start to re-express ICAM and WCAM, so that will recruit more T cell infiltrated. A little bit beyond this is because we originally identified PAC4 as an endothelial cell plasticity regulator. We will see this endothelial cell, once we treat them with PAC4 inhibitor, they can really beautiful normalized vasculature. So this is not like a traditional anti-angiogenic treatment. They decrease vascular density, but here you can see the vascular density is almost same, but you can see the structure is completely switched to a tree-like structure from the chaotic like mass organization. So and meanwhile, we also see the hypoxia is robustly reduced. And then based on this study, we are thinking about, so maybe they will provide the opportunity for CAR T immunotherapy. So again, so once we treat the CAR T cells, along with the our model with the CAR T cell, along with this, did not see really good like a therapeutic phenotype. But once we combine PAC4 inhibition with this CAR, and then we can see much improve the therapeutics. So one thing I've got to emphasize is why we did not see a so robust phenotype, because we only choose a CAR T cells. This model only 40 or 50% of these tumor cells are expressing the CAR antigen like EGFR3. So this is exactly happening in clinic. So in our human patients, so that's why we did this on purpose. So we put all of them together. Turns out this is a really, I would like to say, complicated pathway behind this phenotype. So actually PAC4 can drive a transcription factor activation. It's called MEF2D, and this transcription factor will bind to the promoter of ZEP1. So ZEP1 is a famous EMT, repress, and will bind to the Claudin 14 to block the Claudin 14 expression. So this will will let this and cis and interaction become much less, and then they will dissociate from each other, start to sprouting. And meanwhile, PAC4 drives slug expression. Slug will bind into the ICAM, WCAM promoter and then block their expression. So that's why T-cell adhesion also is also inhibited. So once we block the PAC4, and hopefully we can reprogram the endothelial cell transcriptome and let good things happening and then promotes T-cell based immunotherapy. So that is something we're trying to work with. So that is the original idea how we can really do something. I mean, for example, like genetic reprogramming and T cells. And then during that time, we're also thinking another question is, so because endothelial cells also have a really robust, metabolic activity. So is there any possible this phenotype is also associated with metabolic change? So we just starting to look at a kind of like a screening. So basically, again, we isolate endothelials from multiple different patients, and then we did like a mass spectrometric-based metabolomic analysis. We try to figure out what's the pathway that is metabolic pathway that is altered in these tumor-associated endothelial cells. And then meanwhile, we did the bulk-on-seq for these, tumor-derived endothelial cells to look at the gene expression, specifically look at these metabolism-associated genes. In our human, there's about like 3,000 genes associated with metabolic regulation. So, and then good news is people already did a really nice job to group these genes, and then we know which pathway is associated with certain genes. And once we put all these metabolic data and genetic data together, and turns out very interesting thing happening is, turns out glycine metabolism is the top for the both genetic change and also metabolic change. So this leads us to think about maybe serine metabolism is the major change pathway in tumor and endothelial cells. And then we confirm this, actually turns out the PHGDH, the rate determined enzyme for this pathway is highly upregulated in tumor-associated endothelial cells. So actually, if you look at the pathway, the PHGDH is critical for the de novo serine biosynthesis. And this pathway, it has multiple major functions. First one, they will generate lots of nucleides through the one carbon metabolism. So, and turns out this is very important for cell proliferation. And second thing is they will generate lots of like glutathione, and then everybody knows glutathione is very important for redox homeostasis. So these two things gonna be very important for cell proliferation. So this will easily let us to hypothesis. This is something we just discovered for the oversprouts of endothelial cell, and then eventually drive this abnormal vascular phenotype. So actually, when we look at this, this is something, it should happen to the tumor endothelial cells. We're using either pharmacology inhibitor, WQ, or we're using the siRNA, lockdown, and we can see once we block the inactive of the PHGDH, we can see dramatic reduction of glutathione, and also we can see, nucleotides, biosynthesis. Here we check the carbon flux, so using the glucose like tracing. All of them is done, and then this is highly consistent with traditional idea about the PHGDH role for the cell proliferation. A little bit interesting for us is once we block this PHGD activity, you can see also this glucose activity is also, this is a little bit puzzle for us. And then we'll check, maybe this is due to redox. And then actually this is true. Once we supplement the cells with more, additional glutathione, and then we can rescue that little bit of phenotype. So that means the actually the glucose, metabolism actually for the person, this is something that require this redox balance. So this lets us to think about maybe this is something highly related to the endothelial overgrowth. So actually, once we look at this RNA-seq, bulk RNA-seq data, actually they confirm our idea. So basically here is we take out the tumor endothelial cells and then using the CRISPR-based lockouts, and then we send out the samples for the bulk RNA-seq. And we did not see robust whole transcriptome change, but we can see the majority changing are highly associated with cell cycle and cell proliferation. That confirm is critical role for the cell proliferation. And also we check the single cell RNA-seq data. So with using our own mouse GBM tumor and also human samples, and we can see this once you look at the PhD expression in different like a lineage and the cell, can see this is for the normal and the cell, this is not something associated with this kind of like trajectory, but for tumor cells can do beautiful like a similar pattern. So this is tell us PhD expression is correlates with tumor and the cell proliferation. And then once we using the specific inhibitor, can see and the cells morphology become much normal. Tumor and the cells once we inhibit them or using the lockdown them. So more challenging for us, we got to think about what exactly happened in vivo. So it took us almost 2 years to generate, we eventually generated the Fox, the PHGDH animal. And then we cross them with endothelial specific crane to generate endothelial specific PHGDH lockout or lockdown animal. So we actually, once we test them in with our tumor model, we can see this is a really interesting change of happening to this tumor. This is not like what we have done with this PAC4, like this happened to vascular modification. It's not like what we have done with anti-VEGF, the shut down the vascular. We can see the phenotype turns out is to prune these tumor vessels. So this is exactly happen like what we have seen in our backyards. Like if the sun tree gets like sick, the gardener, the most important for them is cut off the bad branch and then will let healthy branch to grow, and then tree has a much better function. Exactly same thing happened to this tumor vasculature. Once we're using PHGD lockout, genetic lockout, you can see the prune this vasculature to cut off these oversprouting branches and then let these remaining vessels become much more functional. So if you look at the hypoxia, you can see it's original, not original, control tumor always has extensive hypoxia. But if you look at the lockouts PHDTH animal, this hypoxia is much less. And then the tumor microenvironment get completely reconditioned, reshaped, and result dramatic, massive infiltration of T cells. So T cells definitely really need this good vessel to develop the tumor. And also once we reduce hypoxia, T cell become much active. So based on this, and then we'll think maybe they can stimulate the T cell, CAR T cell infiltration. And here is one example we can show is we specifically label CAR T cells with nanoreceptors. You can individually check them, their infiltration and persistence. As you can see here, the PHGD inhibition can stimulate CAR T infiltration, because most likely because the vessel get localized and then also local environment become less hypoxia. Another thing for us is interesting for us is we have seen something different. So because one we will check the additional metabolism because we think this dramatic T cell infiltration, maybe this is only due to the vessel, structural normalization, maybe something more profound actually took place. One thing is maybe they will change the local metabolize. So because a lot of things, especially for the glucose-derived metabolites, for example, like 2-HG and lactate, they can directly inhibit the T cell activity. And the interesting part for us is when we do the comprehensive glucose tracing, we found is once we block the PHGD activity, and then you can see they can robustly reduce the TCL cycle. You can see the alpha KG, malatotes, glutamates, aspartate, all of them level is down. And also very interesting, you can see the 2-HG is down. So 2-HG, everybody knows this is very robust T cell activity inhibitor. So that's why when we look at this cytoph data, you can see this PHGD inhibition, not only they can increase the T cell infiltration, but also they can stimulate T cell activity. You can see this the more CD8, phenotype order, and also once you look at this CD8 T cell, this T cell turns out to express more interferon gamma, granzyme B, and also big T cell become even a little bit more proliferative and then express more Ki67. So put everything together, this PHD turns out is good target, and we'll think about how we can combine this with CAR T immunotherapy. So even the single dose a single dose of CAR T combination with the PEC4, they can kill 10 to 20% of the animal. So we have to see, we have to optimize the condition. We'll see maybe once we optimize condition, for example, like we will give the mice second dose CAR, maybe we'll even see more robust results as shown here. So we can tell you is the CAR T cell turns out has persistent problems. So once we inject them, after 5 days, all the T cells is done, and maybe at that time we should think about the second dose of the CAR. So currently we are doing these experiments. So put everything together, we'll see. People always thinking about the vasculature. The role of vascular in the tumor is they provide oxygen and the nutrients for the tumor growth. Perhaps they will generate some like a paracrine effect and then stimulate the local immunity. But here we emphasize vascular actually has multiple different roles for the tumor immunity regulation. First thing is they control the T cell activity because they have robustly regulate their expression of the adhesion protein like ICAM, WECAM, and they will control the T cell adhesion and the infiltration. And also their delivery function turns out is very critical for any immune cells, especially for the lymphocytes get into, recruitments. So this lets us to think about we can reprogram the endothelial cells and let endothelial cells become like more function to degrade the T cells, more efficiently. And one thing I got to emphasize is the key regulator we think behind this is endothelial plasticity. So one lesson we can learn from the embryo development is so when our baby in their mother's like womb, they have really robust regulation mechanism. So for example, like during that time, and the cells express very little of adhesion protein ICAM, WECAM, because stimulate local immune response, because this is completely two different bodies. So that's why endothelial at that time they on purpose to shut off the loss of ICAM WECAM expression. And maybe tumor, we're thinking tumor maybe hijack this mechanism and then make these things happen again back to the embryonic environment, and then they will locally shut off lots of T cell based pro-immunity activity. So we believe the endothelial plasticity is the major regulator for this vascular mediated T cell immunity. So that's the first part. We think endothelial cell, local niche will generate a kind of very important environment for T cell activity regulation. And second thing, we think endothelium itself also can actively interact with myeloid cells. So that's why exactly currently our lab is working with. They can interact with macrophage, I just mentioned, they can secrete IL-6 to reprogram macrophage and then let this vascular unit become immunosuppressed to block the T cell infiltration or maybe block the NK T cell activity. And second, since we're working with, they can interact with dendritic cells and MDSC, they can block the local immunity. And the final part we will emphasize is they also has lots of metabolic regulation out there. And the CD cell itself can secrete lots of immunosuppressive metabolites, for example, like 2-HG and the lactase and locally block the T cell activity. So put everything together, we believe the vasculature is a very important regulator for, tumor immunity. So I think according to trend, so the time should be 45 minutes exactly on time. So I will take 1 or 2 minutes to thank our lab. Just lots of work was done by talented graduate students and posters, and we'll have lots of good collaboration within UPenn and also, other university, and this work was supported by several NIH grants and foundation grants. I would like to stop here and then happy to take any questions.